Hey there.
Hi, how are you?
Thank you for meeting me at this time. Uh,
Sure. Hold on, I'm just trying to sign into my account.
Did you end up taking a new test?
No, I'm doing it tomorrow.
Got it.
All right. Um, I've been going over, I was doing a lot of psych and soc yesterday. Like I went back over all the brain stuff, the different parts of the brain and then some of their functions and then I don't know, like just things that I thought would be a little bit more high yield. I went over like biochem for the test. We can like keep going over questions, but I've been doing the blind review thing that you suggested, and it is very useful. Like I've noticed when I'm not looking directly at the answers, I'm thinking more about like how to go out with the question. And I do feel like the strategies that I'm using are changing, and I'm like, okay, this was the stupid thing. And I shouldn't be like guessing blindly. Like I should be thinking things through because a lot of the times I do know what is going on in the question.
Yep.
But yeah. I
Got it.
don't know. We could just do like a little practice bank
Uh-huh.
like we did in the past.
When you say a practice bank, what do you mean?
Um, the AMC, like the subject, or we can keep going over the test, like whatever you think is best. I
Uh,
don't know, maybe doing practice.
yeah, yeah. Anything else from that test that you took that, you feel
I feel like I conceptually, like I know amino acids, I know their structure, but I noticed that like I forgot which ones were salt bridges, and so immediately I just wrote it down on the paper and I like took a guess. Then when I thought about it, like salt bridges has to be something that's like charged. So then I was thinking about like negative or like positively charged amino acids, and then like the answer choices become more clear once you like start thinking things through. So I really think that that's my biggest problem that I've noticed this week is just that I'm not like I'm being a little bit too like fast to say that I don't know something.
Yep, yep. Yeah.
But
So,
yeah.
uh-huh. So yeah, if you still have questions from that test, we can go over it. If not, we can do something new from the
Um, like on the topic of amino acids, there was a question that was like, well, I wrote it down. I don't know if this was the question, but like what is a beta branched amino acid?
Oh,
Like I just like didn't know what that meant.
uh-huh. Um,
Probably somewhere from biochem, but I'm not sure.
yeah. So I think that will be involving amino acids that have like a branch, meaning like let me wait, do you mean branched chain amino acids or
The question specifically was saying like beta branched, which like alpha or helixes or beta pleated sheets? Like, is it that? But then beta branched.
okay, okay. Yeah, so that's okay.
Honestly, I wasn't sure.
Yeah. So that's going to be um an amino acid that has a branch in it as opposed to like an amino acid that so like for example, leucine, right,
Mhm.
would just be like
A normal one? There, it doesn't have branches. It's like CH2, right? Is the end.
um
So if anything, it would like have an H bond on the end.
so let's see. So NH2, it's good to know like how to draw the generalized um amino acid backbone like this, with this being the R group.
Hmm.
Now, for let's say for leucine, this would be something like, so I'll draw this again. And for leucine, I think it would be like this.
For leucine, they all go on top of each other. It's like CH2, CH2.
Um,
Get branches.
yeah, so this one over here would be a CH two, and then these would be two CH3s.
Mm-hmm.
But you can also have something like this. Like this for isoleucine.
Isoleucine.
The alpha and the beta just refer to like what's closest to the carbonyl group is going to be the alpha, and what is next closest will be like the beta. So for here, like this carbon here that's attached to like the amine and the side chain is the alpha. But you can also have something like this, where that's alpha and this is beta. Right? So if we look at leucine, this is the alpha, and this
The beta.
should be the beta. For here, this is the alpha, this is the beta.
So wouldn't,
Mm-hmm.
like, I mean, with the exception of something like proline,
Mm-hmm.
don't most amino acids have beta branches then?
Well, if it branches out from that beta carbon, meaning like two separate things instead of like two of the same thing, like for the leucine here, we have the branch attached to the alpha. Like this is the branch, and it's attached to this alpha carbon. Whereas for isoleucine here, you have an alpha carbon here, you have the beta carbon here, and the branch, which is this,
Comes from the beta carbon. Okay.
yeah. So this would be like, isoleucine would be,
Wait, but for the leucine, it's also on the beta branch.
well, the branch is this entire thing.
Right, but that's where the beta is. It's on that like point.
So it branch, so the branch here is this entire thing, right? Whereas for here, it's this entire thing.
Okay.
So this entire, branch is attached to this alpha carbon. But here we have like a new branch, right? Where it's attached to this one, to the beta.
Hmm.
So basically, maybe I could put it draw it in a different way. So um let me draw it like this, like COOH, and then the amine here, and then we can draw CH, CH, CH3, CH3, and then I could also do this. like that. So here we have this as the
Alpha.
oops. So sorry, that would be alpha. This would be, whoops, not that one. That one would be beta.
Okay.
So
So
the
I've heard of that, like alpha carbons, beta carbons. It's just that, and then it's branches that occur on that carbon.
yeah. So yeah, so if we're counting the number of like the carbon that's closest to like the high priority stuff, like the carboxylic acid or the amine,
Mm-hmm.
the first carbon that's attached to the carboxylic acid and the amine is going to be the alpha. And then as you go outwards, it'll be like beta, gamma, etc.
Right.
So yeah. So you're essentially going to have, so the question was for beta branch. So you just want to look for an amino acid where the branch is connected to the beta carbon, right? And so
Right.
here we can see that this branch, or rather this branch, I think it's connected to like the gamma carbon here. Whereas here, this branch, it starts to branch out once it's from the beta carbon. So essentially you want um yeah, you want the branch to be attached to the carbon that's the second carbon after the carboxylic acid group.
Okay.
Like basically,
Uh-huh. That makes sense.
if it had something like this, or if I had something like this would be the alpha, this would be the beta, this would be the gamma.
Mm-hmm.
So so yeah, I think what they would be, or what an example of the beta branch would, the only one that I could really think of is leucine. Sorry, isoleucine compared to leucine.
Honestly, like I wish I remembered like where the question was and I would show it to you. Like I remember that
Mhm,
that's something that raised up a question in my mind and I was like, honestly, I don't know.
mhm.
But this makes a lot more sense, so
Yeah.
I'm glad I asked.
Yep.
So should we do some practice?
Yep.
Okay. Eh,
Yeah. Also, are there any like subject areas or topics that you feel not the best like in?
Um, yeah. Could we do this was also a question that I had, like uh electrophiles. I wrote down electrophiles SN1 and SN2.
Yeah, I have a PDF in the PDFs folder
Mm-hmm.
called like organic chemistry. If you can open that up, I can go through it. It'll be called organic chem intro. It'll be in the PDFs folder.
Yeah.
Yep, that's a good resource right there. But yeah.
Um, where do I start from? O P
Oh,
and
so it's in the shared MCAT folder.
is this not it?
the shared MCAT folder on Google Drive.
Just going through our text, I thought that this was the, oh. Sorry, I opened the wrong one.
It's okay. Oh yeah, it'll be here. I can send a link.
It might have been in the email and I went to the wrong thing.
Here, I put the link in the chat.
Okay. I probably have this open somewhere on my computer, honestly. Okay, so
Yep. So yeah, it'll just be called Organic Chem Intro. Yeah, PDF. But you had it before. Yeah.
mhm.
So yeah, okay. So basically, if I like the best way for me to summarize organic chemistry is how electrophiles and nucleophiles react. And I'm just using this kind of like notation with the electrophile being a capital E with a plus sign and the nucleophile being a capital N with a negative sign. That does not mean that an electrophile needs to be positive, and it doesn't mean that a nucleophile needs to be negative.
Okay,
It does mean that an electrophile should have a partial positive at the very least, and the nucleophile a partial negative at the very least.
right. Okay.
And the electrophile is a Lewis acid, which is an electron pair acceptor, and a Lewis base is the electron pair donor.
Mm-hmm.
And what do I say is the most common nucleophile you'll see on the test?
Not sure.
It'll be the hydroxyl group.
Oh, okay, that makes sense.
What about the most common electrophile?
Like oxygen?
Kinda. So let's say this is an OH group,
Yeah.
and then let's say we have a carbonyl group.
Okay.
So the hydroxyl group is a great nucleophile because it has two lone pairs that it could donate. And the carbonyl group will have a polarity, right? A dipole,
Yeah.
making there be a partial positive and a partial negative, right?
Mm-hmm.
Where would those be?
On opposite ends, probably.
So tell me where in this molecule.
So oxygen usually has a charge,
So,
and then carbon.
huh? Tell me more about what type of charge.
Oxygen has a negative charge.
Yeah.
It's more electronegative, like as far as carbon, it's not so electronegative, so it'd probably be like at least partially positive.
Yep. Which one would be partially positive?
The carbon.
So
Yeah.
we'll attack that, forming a bond with it, momentarily removing this double bond to oxygen to be on the oxygen itself, to be an O negative. But it'll come back to recreate that double bond, and then it'll kick off the leaving group. Right?
Mm-hmm.
So remember that, like, when you see hydroxyl groups, think nucleophile. And when you see carbonyl groups, think electrophile. And we can bring this together with kinases because kinases use which amino acid? Like, so what are kinases?
Um, Something to do with alcohol, right? Or that's a ketone.
Um, well, so kinase is an enzyme, right?
Mm-hmm.
And
Phosphate
something
groups.
with that. Do you know
Kinase. I
different?
was thinking of ketone with alcohol. Kinase is like ATP.
So, do you remember like the different enzyme classes?
Probably, yeah, I think so.
All right, tell me.
Like what they do.
Yeah, and what the classes are, like the generalized classes.
Well, there's like ones that like play a role in RNA stuff.
Uh-huh.
I don't really know if I know specifically what you mean by classification of them, but
Uh-huh. So
like I have a pretty good idea of enzymes, I feel like.
so I put this in the chat. See if you can like share like that as well. I think if you like share your screen instead of just the
Oh, okay, yeah.
yes.
I
Okay.
do. Okay, oxidative
Yeah, yeah.
reductase. Yeah, I do know these things.
Good, good. So oxidoreductases are ones that do redox
Hmm.
and you know,
Transferase, like they transfer functional groups.
yeah, so the type of oxidoreductase you'll see a lot is, well, you can look at the examples. Which one do you see the most?
Honestly, I see a lot of these.
You see,
Like isomerase is super common, I feel like.
oh, sorry, I just mean,
Yeah. Redox reactions happen a lot.
so
Hy
oxidoreductase,
Hydrolysis
so
for sure.
I mean, so for oxidoreductase, these examples, which ones do you see the most?
Um, Dehydrogenase.
That's what you see a lot in metabolic pathways, right? So
Mm-hmm.
that's an example of an oxidoreductase, which is an enzyme that participates in redox, right?
Mm-hmm.
So typically you'll see with dehydrogenases something like NAD plus turning into NADH or NADH turning into NAD plus
Yeah.
or FAD, FADH2 and stuff.
Mm-hmm.
So that's those are dehydrogenases, which are types of oxidoreductases, aka redox. Um, then we have transferases, which just transfer some type of functional.
Functional groups,
Yeah.
usually.
Now, they didn't write this, but the most common one you'll really see is the kinase.
So
Okay.
it's a type of transferase. Okay.
Yeah. Because what people usually get wrong is because people usually think that a kinase would just phosphorylate something, but it
Mm-hmm.
just as well dephosphorylate something.
So
Like,
it could transfer it either way.
yeah, that's why it's just a transferase, right? Like
Okay.
glucose 6 phosphate uses hexokinase or in the liver glucokinase. And so that transfers a phosphate group from ATP to glucose to make glucose 6 phosphate, right? Right. Um,
Yeah.
but then you have pyruvate kinase that takes a phosphorylated version of pyruvate and dephosphorylates it, right? To give you just pyruvate from like PEP. So
Yeah.
that shows you that kinases don't just add, they can also remove. So that's why they're transferases. They just transfer phosphate groups. Well, a transferase will transfer a functional group. A kinase is a special type of transferase that transfers a phosphate group, and it needs to use, in order to assist it, it needs to use three types of amino acids. Which three?
Three,
Like it one of
like for these kinds of things, it's usually like cysteine, serine. Like those are the common ones that get used in
so you're
enzyme.
half correct.
Which one was wrong?
Think
And
about
cysteine is probably right. I feel like the sulfurs are
what's
used a lot.
think about what's closest to what I just went over with the hydroxyl group.
Okay.
So you said serine. What side chain does that have?
Serine I'm wrong about because I was thinking,
Mhm.
uh, it's for sure,
So
honestly, like
yeah.
I know, I don't know, I just feel like serine is commonly like used for things like this.
What
And
what
then
what what can you tell me about the structure of serine?
what's polar?
Um, anything else? Like what?
Like what molecule wise? Um, there's no, well, I was thinking of, okay, I brought cysteine into it, but that one has a sulfur spray. This one doesn't. It's just like OHs and CH2s.
So you mentioned something right there.
But it has the OH, so it can like lose a hydrogen.
So if it has an OH, right?
It can be reduced or oxidized.
Well,
Is that where you're going?
so what reason did I bring up OH here?
Well, the charges of the OH group.
Well, so I was trying to say the most common.
The most common, you said OH was a nucleophile.
Yeah.
Or no, electrophile.
Which one?
Electrophile.
Well, what did I say an electrophile was?
Electrophile has a E negative, right?
Um,
Oh no, I flipped it. It's E positive and negative. So it's a nucleophile because it has the partial negative charge.
so let's think about it like this, right? So remember, nucleophile, it doesn't necessarily need to have a negative charge, right? But it's a Lewis base. And tell me what a Lewis base does.
Honestly, I'm not sure. Like I know it has something to do with structure,
So
like Lewis structure, the Lewis base, and it's just the definition of it is the like what a base is.
so
It's
so if we are talking about acids and bases,
oh, it's like a electron donor versus electron receptor. That's what Lewis is. It's just that's what the like the definitions that it gives. Okay.
yeah. So you could see this hydroxyl group that I drew, right? It has two lone
Hmm.
pairs, right?
Yeah.
And that's what it's going to donate. So
Right.
hydroxyl group, again, is the most common nucleophile. And a nucleophile is a Lewis base. So it's the most common nucleophile, aka Lewis base, you'll see on the exam. So when you see the OH group, I want you to be thinking automatically nucleophile
Lewis
is going to
base.
react
And
with things, right? So
yeah.
we have that. And then I said this carbonyl, something with a carbonyl group would be the most common one.
Most at least partially positive. Carbonyl is oh, so that's what you were saying. That it's a base and the other one is a Lewis acid
Yep. And a Lewis,
by revert. And acidic would be more negative.
so a Lewis
So the
acid is a what? Is a electron pair.
what?
Sorry,
Like
so
which one pairs?
so um so the this carbonyl molecule that I drew is an electrophile, which means it's a
Well,
Lewis.
that would be the like electron receptor
So it would be a Lewis
acid.
acid, right? Because it's
Yeah.
accepting a lone pair, right?
Yeah.
Good. So remember hydroxyl group when you see it, right? Think
Lone,
nuclear.
sorry, Lewis base and nucleophile. So that makes the other one an electrophile and a Lewis acid.
Correct. Now, something that looks similar to this carbonyl is a phosphate. You should know how a phosphate looks like, right? So, like, for simplicity's sake, I'm just going to write like two phosphates here by just writing P with a circle in it.
Mm-hmm.
And so the same type of notation, so for ATP, right? The closest phosphate to because there's going to be like a ribose here and then adenine here, right? It's kind of hard to see. I'll make it like this. With an adenine here, right?
Mm-hmm.
So, so this is ATP with the first phosphate that's closest to the sugar as the alpha phosphate.
Okay.
Next one beta, next one gamma.
Yeah.
So an OH group, so you see how the phosphate is similar to the carbonyl in that it's bound to an electronegative atom like oxygen. And if it's bound to something that's so electronegative, that electronegative atom will pull electron density away from that phosphorus atom, right?
Mm-hmm.
So and this phosphorus atom is bound to 4 oxygens, so it's really so it really has its electrons taken away, right? And that's not, it doesn't want to be in that because it's not really equal. So OH comes in and nucleophilically attacks that just as it would nucleophilically attack, because we have a partial positive here, just as it would nucleophilically attack a carbonyl carbon. All right. And
Mm-hmm.
as it does this, it'll move the electrons from this double bond up to the oxygen to give you an O minus, but then it'll come back, recreate that double bond, and kick off this leaving group, right? Which would mean that basically, this bond to the other phosphates breaks. Over here.
Because it's so far back, so it's weaker.
Oh, sorry. Oh, so just like here, right? It's attacking, the OH is attacking the carbonyl
This
carbon.
is the thing where like it takes the hydrogen from one side and like something else from a different one, and this is like it forms an H2O.
So,
That's like if it leaves, and then if it comes back, it could like ligate
uh-huh.
the ends or like jumping to something else.
Yeah, so I have okay. In the PDFs folder, click on open up like I have a PDF that's just called Kinase STY.
The different one.
Yeah, see?
My like computer stuff typing. I'm like stuck in.
Yeah.
Do you see the plus sign? I don't know what that is.
A plus sign? I see like
I'm drawing. I'm annotating something.
I think, yeah.
Okay. 101.
Oh no, just kinase. That's weird. Oh yeah, here. STY over here. Down here.
You
Down here.
see it? Oh. Okay.
So here we go, right? So a kinase helps yeah. Kinase helps.
That's it. There's nothing above it. It's just this picture.
Yeah. So kinase helps the hydroxyl group on STY, which are the amino acids, serine, threonine, and tyrosine. What they have in common is that they have hydroxyl groups. So the hydroxyl group nucleophilically attacks the gamma phosphate group, which is the third phosphate of ATP, resulting in the amino acid residue acquiring a phosphate group. So we can see here that we have this OH group, which is from STY, serine, threonine, tyrosine. And you can see that this electronegative oxygen with the lone pair attacks attacks the phosphorus atom here, and then it will break off the phosphodiester bond to the next phosphate to give you this, which is the amino acid that could be ST or Y, that's now phosphorylated. And you get ADP over here. So this is a very high yield type of question that you'll see them at. They'll ask you something like, I just saw a passage that was like amino acid 19 of light chain 20 gets phosphorylated. What type of amino acid is amino acid 19? It would have to be ST or Y.
So it's serine, tyrosine, and threonine?
Yeah, with the T being threonine.
Yeah.
So, and the Y being tyrosine. So that's, yeah, so you're essentially having your nucleophilic hydroxyl group attack the electrophilic phosphate atom of ATP, let's say, and it can phosphorylate that STY. So that's how the kinase will transfer phosphates to and from things.
Okay.
Yep.
Sounds good.
Yeah.
All right.
So, yep, you could bring up that the enzyme thing again, like in the chat. Oh, it'll just be in the chat.
You don't think it'll still be no.
Oh no, because this is just going over the Google Drive, but I put the enzyme thing, the picture in the chat.
Oh, this one.
Yep. So yeah, so kinase is going to be this type of transferase. And then we have hydrolases, which are just the hydrolysis. And these are like protease. They could also be like nuclease, things like that.
I didn't realize that lyase and ligase were different. Catalyze the addition of groups to double bonds. Okay.
Yeah, they can be kind of confusing in that they sound very much the same. But you can kind of remember what we talked about Y kind of being something that can like break something. I mean,
And ligase is joined together.
yeah. The ligase could also add things to double bonds or form double bonds, but when you do that, you're removing stuff anyway. So like decarboxylase would remove CO2. And then you have isomerases, which just move things around. Ligases connect things together. Translocases can move things like across membranes, kind of like when we went over like transporters and stuff. So yep, so there you go. So those are the enzyme classes. So there's that. And yeah, we can go back to that Orgo PDF. Yep. Good. So, okay. So I then talk about nucleophilicity, and it roughly parallels basicity, but bases are defined by abstracting, meaning removing a proton, aka the Bronte-Lowry definition, whereas nucleophiles are defined by attacking partial positive, typically carbons, but in the example I gave with phosphate, it's a phosphorus that it attacks. Differences are due to these factors. And then I just go over like basicity and nucleophilicity, some more differences between the two, but we can go back, go down to just this overall nucleophile strength. And then how to deal with like whether it's SN.
So it's Funkelbush in the opposite order. Or, like, essentially.
Kind of.
There's like SHCN. Oh, I guess not.
Yeah. Because there's different factors like polarizability, which is if something larger, if you can stabilize a charge over a larger distance or area, it's more stable. So the size is going to matter, some other stuff, but yeah, here what we first want to do is ask, does the nucleophile attack first, or does the leaving group leave first? Right? So if
I
the
feel like um wouldn't but something would have to attack to make the leaving group leave. Like it wouldn't just dissociate on its own.
yeah, it could be, you'll always have a solvent anyway.
Uh-huh.
So it could just be that. All right.
So it's in the solvent. I mean, I think that for the reaction to start, then something like the leaving group has to leave. Like that's the first step of most reactions is like something leaves and allows the like catalysis or whatever to start.
Yeah, so we can have okay, if the nucleophile attacks first, it'll be SN2. If the leaving group leaves first, it's SN1. I'll go more into that. And if there's a high temperature, there could be elimination. So if it's high temperature and an acid catalyzed, it'll be E1. If it's basic, it's E2. So okay, I go more into SN2 here. So strong, if you have a strong nucleophile, and what it would do is it would do a backside attack to a less hindered electrophile. So that means primary is more likely to be SN2, followed by secondary.
Oh, okay. I remember this. Like and it's the opposite order for um SN1. And isn't SN2 more common even though it's called 2 instead of 1? Or there was something where it's like it usually happens that way.
Uh, I can't answer like if it's more common or not because they can ask you anything. Um, like one of the questions we did go over once was like which of the following would be like involved in an SN1 reaction.
Probably.
But so what we have here is that if you have an electrophile that is not very much hindered, your strong nucleophile will backside attack it, forming a high energy, which means unstable, 5 ligand transition state, which would have a shape of sp2 trigonal bipyramidal. And this is followed by a single enantiomeric product with inverted stereochemistry. So
Okay.
you scroll down, you can see what this would look like. So we see that we have this molecule here, right? And is this molecule that I drew primary, secondary, tertiary?
Primary.
Close. It'll be well, all right, we can say that it's primary.
Well, I'm going off of like if you look here, the
Mhm.
it's attacking it. The nucleophile is attacking it, and that's starting the reaction. So if we looked at like what SN2 was, the less hindered. So it goes in this order where it's like 1, set 2, 3.
Mhm.
So I don't know. I assumed that like it would start since the nucleophile is attacking with 1.
Yeah. So I think uh it's just because I put a leaving group there, but yeah, so it's going to be like let's say that it's because if this leaving group was just another H, that would be technically primary, but it's still fine as long as it's not like tertiary, right? So let's just say it's primary. So we have basically our molecule here, and we have H is going into the page and out of the page. And this is not very sterically hindered. We don't have many R's here. We have H's. So this nucleophile does not have
Is
any.
the second one tertiary? Sorry to interrupt. Because like the leaving group is now leaving.
Well, no, because
That's not how the order goes.
yeah. So let's consider this to be primary, right? With the leaving group, not like if the leaving group isn't an R group, then it's not going to be count it anyway. So let's just, yeah, this is pretty much primary. But essentially what I want you to see though is that the leaving group is on one side, and then the other side has the R group and the two H's. So we can say that that's the back of it.
Mm-hmm.
Nucleophile will attack the back of it. That's the backside attack. And since it's just hydrogens and of course one R group, there isn't much steric hindrance that would block that nucleophile from attacking.
Okay.
Right? So it's gonna attack, and we get this transition state. Now a transition state, what can you tell me about that compared to, let's say, an intermediate?
Isn't it where like the energy isn't exactly where it's supposed to be? It's something with like charge and transition state.
So the in a reaction diagram like this, right, where this could be reactant over here and this can be product over here, the hump, right, that energy barrier that you need to overcome to make the reaction go forward, right? That's the energy of the transition state, because the transition state is the most high energy form of a reaction.
That makes sense.
Yeah. And
It has to overcome. Okay.
yep. And so we see the transition state here, and what we see is that so the R and the two H's here are like angled like towards the left, but in this transition state, it's almost like they're flat, because what you have is the bond to the leaving group is being broken at the same time that the bond with the nucleophile is being made. Okay.
Okay.
So like, let's say my fingers are like the R and the H's,
Mm-hmm.
and what will happen is the nucleophile would come in, and as it comes in, it'll cause
To flatten out.
yeah. So it'll go like this, and then the bond to the leaving group, when they're both equal, it'll be flat. But then as the leaving group leaves and the nucleophile comes in, it will, I mean, I can't go move
I
it,
understand.
but it'll go like that, right?
Okay.
So the transition state is when you're simultaneously making the bond between the electrophile and the nucleophile.
And the leaving group is leaving.
Exactly. And that's why it's this sp2 trigonal bipyramidal shape, which, by the way, is like this, like that, and like that. I don't know if that's kind of easy to think of, but whoops. Something like this. I don't know. It's kind of like, because I'm making like this triangle here and then that and that. So it's basically a pyramid with a point on the bottom as well,
Okay.
right? So that's the sp2 trigonal bipyramidal as the transition state shape. And then when the leaving group leaves, it will invert the stereochemistry of what's left. So you have this as your product, and then the leaving group is leaving, forced out. And yeah, we get our single enantiomeric product with inverted stereochemistry. So, if this was like R or something like that, this would have to be like S, right?
Mm-hmm.
Okay, good. So this happens in one step. So it's second order kinetics. And that's a weird thing, because it's like, why is it called SN2 if it's one step? Why is SN1 called SN1 if it's 2 steps. It's because of the kinetics, the rate law. If it's second order, it will depend on the concentration of the nucleophile as well as the electrophile. So the rate can be expressed as, K, that rate constant, times the concentration of nucleophile times the concentration of electrophile. Right?
Okay, that makes sense.
And yeah, it happens fast. It prefers polar aprotic solvents. And some, I guess, could see that this transition state exists. Now, if we go down to SN1,
Right here.
now if the electrophile is sterically hindered, so if it's
Then 3, 2, 1.
and then it'll be a weak nucleophile, has to wait until the leaving group leaves. So it's like the nucleophile is like a robber or something, and if the electrophile is alone, the robber is gonna be like, hey, give me all your money. But if the electrophile has like friends hanging out with him or her, the robber, the nucleophile, is like, oh crap, you know what? I can't do anything. I need to wait until the friends leave, and then I can come in. Right? So I drew a similar molecule here, except now I made it tertiary because there's, instead of it being an R and 2 H's, I made it 3 R's. Right?
Okay.
So so yeah, go back up here. So okay, for SN1, right? You see how this is tertiary now
Yeah.
because of the 3 R groups? Yeah. Are you good?
Yeah.
Okay. So look, there's 3 R groups, right? So a nucleophile is not gonna want to do anything, right? Because there's too much steric hindrance. Okay. So what it has to do is it has to wait for a leaving group to leave. So that's what happens. Leaving group can leave, and you'll be left with this. What is this here? Is this a transition state?
By effect, wouldn't it be?
What's up?
That's how it went in the SN2 reaction. So it should be, right? This is like the leaving group left, and now like the reaction can start. So this is when the nucleophile attacks it. And
So let's compare it. So if we compared it to the previous one, right? The previous one, the transition state was simultaneously breaking a bond as it made a bond.
right. But here it's not happening simultaneously, so it wouldn't have a transition state. But it still, don't all reactions have some sort of transition state?
It will.
Whether it's SN or SN2.
It will. But if you can, so a transition state cannot exist on its own.
Okay.
So so there will be a transition state to get to this point, but this can live on its own, right? Because it has this positive charge. Right? So this can exist on its own because it's just a carbocation. Right? And if it can exist on its own, we call it just an intermediate. Right?
Okay, yeah.
And that's another reason why tertiary is preferred, because it's preferred because tertiary would give you an electrophile that has steric hindrance. But it's also preferred because when the leaving group leaves, the tertiary carbocation is more stable than secondary or primary because it can spread that charge. Okay. Right?
Yeah.
So
Sorry, I'm like trying to process.
it's okay.
I understand. Okay.
Yeah. So over here in this reaction diagram, I wrote it with R being the reactant, and then transition state up here, it does exist.
There's two of them,
Because first
and
is
then the intermediates between them. That makes sense.
exactly it's lower in energy.
Right.
Because the first transition,
Nothing can be as high as transitions.
yeah, the first transition state is the leaving of the leaving group leaving.
Mm-hmm.
The second one is
Second one's the nucleophile attacking, but because it's not together, then there's the two separate steps.
exactly. So at this carbocation intermediate step, the nucleophile can attack from either direction. Like it can attack from the front, it can attack from the back, and it can do that equally. So instead of it inverting the stereochemistry, it will form a racemic mixture of half one type and half the other. So basically, the weak nucleophile will wait until the leaving group leaves to form a 3-ligand intermediate. And then the nucleophile can attack from either side of the planar, the intermediate here is planar, carbocation intermediate, so it forms a racemic mixture. So it can attack from either sides. Now, since this only depends on the substrate, meaning the electrophile, like it entirely depends on the electrophile letting like the leaving group leave. The nucleophile is not having any effect there. So the rate is only determined by the concentration of the electrophile. That makes it first order kinetics, which means that's why it's SN1.
Okay.
Yep.
Sounds good.
Yeah. And then after that, I have some stuff on like E1 and E2. So basically E2 is if you have if you have a bulky base, examples are LD, like tert butoxide is usually the example that they make, which is that, right? So yeah, if the base is bulky, the energy for an SN2 transition state is too high, so it must be overcome with high temperature. When that happens, the base will remove an alpha hydrogen to force a leaving group out, which will result in the creation of a pi bond, aka a double bond, that has to be anti-periplanar. So I drew this over here. And you can see if I were to draw like, I forgot what it was called, like Newman maybe or something, but like H here and then BR here, right?
Mm-hmm.
Anti-periplanar. So like the base can remove the hydrogen here. That will cause a double bond to form here as this bromine leaves to give you this double bond.
Okay. Sounds good.
Yep. And then E1 is also high temperature, but if you have an acid that catalyzes it, then it protonates the leaving group. That can cause the leaving group to leave, leaving you with the carbocation intermediate, which can shift. There could be hydride shifts here or methyl shifts. But yes, you have another pi bond or double bond form. And then I have some other stuff here that you can go over with like how to tell. Right?
Okay.
So yep.
All right. This is like a really good PDF. Wow.
Thank you.
You like
Mm-hmm.
in depth, but not like super long.
Yep. Yeah, I think if you can explain something simply, that shows that you understand it. well.
Okay. All right. So this no.
So, yeah, we could use stuff from that section bank.
I don't know where it's still open. Okay. Right. Should we do biochem maybe?
Sure.
Should I do it with Jack Weston?
Oh yeah, because it'll just be in a nice format. Yep.
I know. Okay. Should I read it?
Uh, did we do this before?
I don't think I've done biochem with you. We
Okay.
only did chem phys.
Mm-hmm. Okay. I was gonna see if there was anything that would be closely related to things that you may get wrong, but
Oh, well,
we can.
I didn't do great on the biochem test that like the portion section of the test that I took. So maybe I don't know. I went over the questions that I got wrong in that section though, and I like that's what I was telling you that a lot of it was
Mhm.
mistakes that weren't necessarily knowledge based or well they were, but it was just that like I immediately said, "Oh no, I have no idea what it is," and I should have like taken a closer look and I would have been able to figure it out.
Mhm.
So like I feel like right now I'm just working on not giving up as
Okay.
much.
All right, let's do that.
Okay. Blood flow to the brain is regulated by changes in arterial vessel diameter in response to intravascular pressure, a process called the myogenic response. The MR allows cerebral resistance vessels to achieve a state of partial constriction from which they can dilate or further constrict to alter blood flow.
All right, what
Vascular,
are they saying there?
what?
Oh yeah, what are they saying there?
Um, so there's a process called okay, well first they were saying like blood flow to the brain is like regulated by intravascular pressure, so like the vessel either like constricts or like opens up can depending on like the pressure
Do you say that it's regulated by intravascular pressure or
by changes in response to the pressure increase or decrease.
changes in what?
Changes in pressure.
Oh, I mean,
Well,
like
it's regulated by changes in the arterial vessel diameter in response to intravascular pressure changes.
good. If we had to assign one to be a independent variable, one to be a dependent, what how would
Well,
you?
blood flow. Oh no, I think the arterial vessel diameter would be the dependent variable because it changes in response to the intravascular pressure, which is the independent one. Okay. The MR
Hmm? Yeah.
allows the vessels to achieve a state of partial constriction. So that's when they're more like compact, in which they can dilate or further constrict to allow blood flow, alter blood flow. So obviously MR has some sort of role in this like regulatory process.
Mhm.
Okay. All right. Vascular smooth muscle contraction is initiated by cell membrane depolarization, which opens voltage-gated calcium channels.
Okay. Now, see any buzzwords here?
Voltage-gated channel for sure.
All right. Anything else?
Depolarization.
Good. Anything else? What's smooth muscle?
Like I'm thinking like smooth or rough,
Yeah, what are
and
they?
ribosomes.
Right,
Well, that would be like in a like ER. Like the presence of ribosomes makes it smooth or rough. I'm not sure what smooth muscle is, but I'm assuming something of the same nature where like it doesn't have.
like a ribosome and endoplasmic reticulum is inside of the cell, right?
Right.
But we're talking about a type of muscle, right?
Right. So I'm saying like on a larger scale of smooth muscle would be lacking some feature or some like extended feature that's present on like a rough muscle.
What do you think about that?
Smooth muscle. Honestly, I don't know what that would be.
Okay, so let's think about the muscle that we do know, right? Um, which is skeletal muscle,
Uh-huh.
and that kind of looks like something like this.
Yeah.
Well, what did I draw here?
This is the overall structure of the. Is it like surrounded by bones? I can't tell. Is the like are the circles the muscles?
So, so this is supposed to be a sarcomere.
Okay.
I put a picture in the chat if you can share that.
Mm-hmm. Oh, okay.
Mm-hmm.
So the bands. Okay.
What can you tell me about this?
So that's like when it's relaxed versus so these are like flexed muscles are more like stretched apart,
Uh-huh.
but the bands
Mm-hmm.
move backwards and then the zone between them increases.
Yeah. Do you remember like the cross bridge cycle?
Probably.
Uh-huh.
Honestly, bio isn't like where I focus my
So, wait, wasn't that your major? Bio?
yeah, but I haven't taken like bio specifically classes in forever. I
Got
take
it.
like plant biology and
Oh, that's
neurobio.
that's so okay. So the basic thing for a cross bridge cycle, which is high yield, is that um so you have so you have actin and myosin, right?
Hmm.
And so in this picture, the actin, which is also known as like the thin filament, is like the blue stuff. And the myosin, also known as the thick, is like the red. When I say
The
red,
red.
this red stuff. And like, let's say this is the actin, and then like, let's say this is like the myosin.
Hmm.
Um, the myosin head could be like 90 degrees or 45 degrees, right? So let's say that. So when there's ADP, for the myosin head, it'll bind to the actin, okay? And it'll hydrolyze the ADP, which will cause the myosin head to go from 90 degrees to 45 degrees. And if it's attached to the actin, when it goes 45 degrees, it'll push the actin, let's say this way, to the left. And if it's attached to this, it'll push the myosin to the right, like going like this to the right. And that's why they will like overlap each other for skeletal muscle contraction. And we call that the power stroke. And then ATP can come in and detach the myosin head from the actin. So you know how animals when they die they have rigor mortis. What is that?
It's when the muscles get like expand.
Oh, what do you mean by that exactly?
I don't know, like something happens in the body where it's like everything is super like expanded and bloated and they like kind of get stuck in the position. Oh, I guess that would be more of like contraction of muscles.
Mhm. Yeah.
Okay.
So when an organism dies, it can no longer take in ATP, and ATP is required to detach the myosin head from the actin filament.
Uh-huh.
That's why when animals die, they don't have that ATP to detach it. So their myosin is permanently stuck to the actin, which is why the muscles are in a state of contraction.
Okay.
Yep. So this is skeletal muscle, okay? And
Mm-hmm.
skeletal muscle is voluntary. We can control it. And these bands, these vertical bands, are what so if I drew the muscles like this, that is what gives it these vertical striations,
Mm-hmm.
thus making it not smooth.
So the presence of bands is what differentiates between skeletal and smooth muscle,
In addition to,
but it still has like the myelin and the actin and the normal like classifications of a muscle, right?
yeah, it'll be in addition to multinucleated versus um uninucleated. So I'm just going to put a picture in the chat that we don't have to go over, but just save it so you have it. But we can see that. So what I sent you, like the skeletal muscle has yeah, is voluntary. It has those striations, but it's also multinucleated, and tubular. So they're like tubes. Smooth muscle, non-striated, spindle shaped, so they taper at the ends, uninucleated, involuntary, and then cardiac muscle is branched and has the striations, and is also uninucleated. Um,
Mm-hmm.
yeah, and smooth muscle and cardiac muscle are involuntary.
Okay.
So yeah, smooth muscle would be like, uh, so like let's say this is your a cross section of your blood vessel. Like this is like the epithelium, but this would be like the endothelium, which is composed of smooth muscle that can contract and relax. So
Okay.
So yeah, so
All
vascular
right.
smooth muscle is the type of muscle that's in blood vessels, and that's what they're talking about.
Mm-hmm. All right.
Mhm.
So
Good. So
over like right here, I think.
yeah, so they said that VSM contraction is initiated by cell membrane depolarization, which opens the voltage-gated calcium channels. So maybe I would write something like VSM contraction proportional to depolarization proportional to
Opening of channels.
yeah, calcium channels opening. Good. Okay.
Okay. Um, calcium ions bond the cytosolic protein calmodulin, which activates an enzyme that phosphorylates amino acid residue 19 of the myosin light chain LC20.
Uh,
Phosphorylation of
sorry.
LC20. Okay.
Sorry. So it's funny because I kind of referenced this, but um what type of enzyme do you think this is? The one that phosphorylates amino acid residue 19?
Phosphorylase.
Are you sure? Maybe something else?
No, because it deals specifically with like a phosphate group.
So other things can do that, like one that we went over recently.
Kinase deals with phosphate groups too, but
Huh? Now,
Is phosphorylation the addition or removal of a phosphate group?
it's addition.
Okay, so then it's probably kinase because that's
So, okay. Now, what do you think amino acid residue 19 would be?
like what specifically 19th? Is it like on a sequence like the 19th amino acid?
So think about
Rotated
kinases and what amino acids they're associated with.
was STP S
Hmm.
TY.
Yeah, good. So right away, we can see that, right? We can
Yeah.
see that this is likely going to be a kinase. Likely, it doesn't have to be, it could not be that. But amino acid 19 would have to be STY if it is a kinase. So good, okay. And they're also saying that the calcium ions bind to calmodulin. So I would say like binds to calmodulin, which means that LC, so LC20 gets phosphorylated, so I just draw like a P with a circle connected to LC20. I'll make it neater, right?
Okay.
What are you thinking?
Sounds good.
Okay. Uh,
And then
yep.
the phosphorylation of LC20 is required to activate the myosin head, which binds to actin. This myosin-actin interaction forms the basis of the cross-bridge cycle, which causes muscle contraction, as in skeletal muscle.
Yep. So that's what we just went over, right, with the cross
Uh-huh.
cycle. So we could say that this kind of out of room here, but I'll kind of write it underneath. Um, would be I'll just write CBC.
Phosphorylation binds to actin,
We're
but it's saying that the phosphorylation of the LC20
mhm.
is required. So does that mean that this would like I don't think that this one's a kinase, but I can't like
Mhm.
is it removing a phosphate?
Which thing? The thought.
Because I'm just now that I'm thinking like this suppress CBC.
Oh,
So if the phosphorylation is required to activate it, so that's the adding of a phosphate group, then to remove it, you would probably need to like suppress it, you would need to remove that phosphate group. So I'm leaning towards C as the answer choice here.
sure, sure, sure. I didn't know you were looking
No,
at.
I just like I was reading I saw the question and as I was reading, I was looking for where CBC would come up and
Mhm.
this is the first time it was mentioned.
Got it, got it. Yep.
Does that like the process of like thought process make sense?
Hmm.
Okay. Okay. Sounds good. So then should I keep reading?
Hmm. I mean, if you want to do the questions first, we could do it.
Well,
Uh,
no, I just like I saw the CBC was on my mind, so
got it.
when it came up, I just wanted to answer it
Cool, cool.
because we haven't even seen cytoskeleton yet. It's like these.
Hmm.
Dynamic reorganization of the actin cytoskeleton occurs during VSM contraction and relaxation. During actin polymerization, monomers of globular actin polymerize into filamentous actin. So globular is like a bigger thing, and then it like polymerizes, it like gets smaller, broken down into like these filaments,
Well, if
smaller
being
pieces,
summarized, wouldn't it make it like,
bigger.
mhm, right?
Like I know Oh, like G protein.
Anything that polymerizes, right, just means that,
Gets bigger. Polymerase is like multiple.
yeah.
Okay, so the monomers polymerize. Okay, that never mind. That actually makes a lot of sense as you read it.
So if I wrote like G-actin polymerizes into,
F-actin,
yep, and then what would I put for this? Or maybe you haven't read that.
depolymerization.
Yeah, maybe you didn't read that yet, but yeah, this last sentence here.
Yeah.
What does that say?
That it like did latch ronculin B. Lat B.
Yeah, that would do that. Good. And also we know G-actin and F-actin should be inversely proportional, right?
Yeah.
Okay.
Okay.
Mhm.
To examine changes that occur in VSM during the MR, researchers exposed isolated rat middle cerebral arteries, RMCA, to increasing internal pressure to mimic intravascular changes that would occur in vivo. Okay. Arterial diameter and the amounts of phosphorylated LC20, a phosphorylated calponin.
I think you. Oh, okay. No, keep going.
Phosphorylation of calponin reduces its inhibition of the actin activated myosin MGATPase, and G-actin were determined in response to three-step pressurization from 10 to 80 to 120 Hertz, right? Or Hg, whatever.
Millimeters for mercury. Yep.
Okay.
Mhm.
All right.
Yep. So for that table, I mean, what I would write would probably be some proportionality stuff as well, like, um,
Fair.
oh, for the table,
Oh, okay.
yeah, like pressure. What would the proportion be to arterial diameter? I mean, just from the table.
As the pressure increases, the diameter decreases.
And phosphorylated LC 20%.
Increases. So it would just be directly proportional.
Yep, and I'll just say until 80 because it flattens out after that, right?
Uh-huh.
All right, and then the
Phosphorylated calponin also increases up until 80.
G-actin,
Decreases, so 1 over G-actin.
which could mean just proportional to F-actin, right?
Right.
And this normalized amount, remember when we talked about normalized, it just means that the amount at 10 millimeters of mercury we equate to 1, and everything else is based on that. So
Okay.
good, awesome. So yep, so we got everything that we need to know. So yeah, let's go for it.
Okay. Which statement regarding VSM function and cytoskeletal dynamics is best supported by the data in table 1? Okay. As F-actin decreases, phosphorylate LC20 would also decrease.
You said F-actin. Did you mean G or?
No, because I'm looking at like B, for example. And since we said G-actin is decreasing, then F-actin would increase as phosphorylated LC20 is increasing. So if one decreases, the other one has to decrease too. So I think B is wrong.
Good.
Vasoconstriction is associated as it increases. Maybe A. I'm not. Because vasoconstriction would affect the arterial diameter. And so as there's going to be less. Oh, wait, no. Arterial diameter and F actin would be proportional. Oh, no, these are proportional because as this one decreases, this one decreases. So if there's more G actin, it's going to be more constricted.
Okay.
So I'm like leaving A for now, but I'm not crossing it out.
Mhm.
Vasodilation does not affect phosphorylation. So if it's dilating, then this one would decrease. Well, yeah. Let's say that this one was, as it's increasing, this one would get even smaller. So I don't think that this is true.
Mhm.
And then arterial diameter reduction is, well, and I think that always is a trick word. So I'm going with A.
All right, but let's just
Okay.
verify
All
that
right.
all the way.
Reduction is always dependent upon increased calponin phosphorylation. Well, I mean, it is proportion. Like it does make sense based off of the chart. Is best supported.
So
Maybe
arterial diameter decreasing from 193 to 175.
did not result in any change in phosphorylated. So that's what I'm saying, that always is kind of the trick here because it isn't always like from the first one, yes.
Yeah, always extreme words you always want to be suspicious about, but I just wanted you to
Right.
make certain.
You just have to validate. Okay.
You know, like the saying, measure twice, cut once.
Never heard it.
That's the idea.
Okay. All right. Compared to control RMCA. Hold an internal pressure out of 120. Application of LAC B to RMCA held. Okay, so applying the LAC B, which I can't see where we talked about LAC B because I know we wrote it down somewhere. Okay, so F actin to G actin. Applying it would decrease levels of F. Just want to cross out the other.
Mhm.
LC20 increased as G went up.
As G went down, you mean?
Went down, yeah.
Going in the other direction, sorry.
So that one's going in the opposite direction, so it would decrease. Decrease in arterial also is correlated with less G, not more. Increased phosphorylated calponin. No. So it's C.
Mm-hmm.
Yep. You're right. This does help a lot.
Yep.
which describes the function of the MR in response to stimulation of the sympathetic nervous system. Did it say something about cerebellar? No, it has more to do with blood flow. Okay. Oh no, allows cerebellar resistance vessels to achieve a state of partial constriction. So not dilate. My brain wanted to go straight to cerebellar resistance. I don't think we know this from the passage. It doesn't say anything about abdominal cavity.
That's something that you should know though. Like,
Well, right, that like if it's in an excited state, so like fight or flight takes away from organs and sends it to the brain so that it's but I don't think that's like
so parasympathetic nervous system would do what D is saying, right?
yeah,
Because it's the rest and digest.
yeah.
So
In the brain to organs. It would go organs to brain in
mhm.
autonomic.
Sympathetic, yeah.
Yeah, okay. Moderates blood flow to the brain under high pressure. So if the pressure was increasing, arterial diameter was decreasing. So was it away from the brain or to? I just,
Mhm.
I guess the like wording of to the brain. Because under high pressure, it definitely does do something, moderates it, like restricts. But I just don't know if it's like to the brain or away from the brain because I feel like normally like blood rushes to the brain. It says alter blood flow. And never blood flow to the brain is regulated, but it never says, oh,
Mhm.
I don't know. I'm going with B.
So let's see what would okay, D would be not it, right? Because D would
Right.
be sympathetic.
Yes.
What do you think about A?
Equalizes blood pressure and cerebral resistance. I mean, those words were directly in the text, but equalizes, I'm not sure that it necessarily equalizes. I think that that would say that there's the same amount of blood on either side, which isn't explicitly said.
So let's think about the aorta.
Okay.
Tell me about that.
Well, that's like pumps blood.
Like from your heart to the rest of your body, right?
Right.
So the aorta should have the highest blood pressure of anywhere in your body.
Right. So why would it be equalized between that and the cerebral resistance arteries?
Yeah. If that was equalized, you'd have like a hemorrhagic stroke of some sort. So yeah, it's not going to be that because the aorta is the highest, blood pressure.
Mm-hmm.
But yeah, moderates is a nice, vague word that means regulate. Doesn't mean, it'll make it less or more. It includes all. So yeah.
Okay. Sounds good.
Mhm.
Do you mind if we stop at 9?
Mm-hmm. Yeah.
Okay. Um, Polymerization of which structural component was analyzed in the experiment described in the passage? Actin. I feel like filaments is a trick. Because it wasn't initially. Or at least it wouldn't. Would it be like intermediate filaments? Is that the answer that they're going for here?
What do you think?
I don't know, because my issue here is that is the wording where
Mhm.
it's saying the monomers of globular, well, whatever it says there. Monomers of globular actin polymerize into F-actin.
Yep.
So if it's polymerizing something, then it's obviously not polymerizing the filament. It's turning into that.
So, um, so from that image that I sent of like the sarcomere,
Uh-huh.
one of the things that I said was that the myosin we consider to be thick and the actin we consider to be thin.
Right. So it would be
Uh-huh.
micro, but it's not a tubule because that's not being discussed anywhere. So it's still filament. But it obviously, like, I knew from the beginning that it wouldn't be like a thick filament because that just doesn't make sense. So a B. Okay.
Yep. Knew it.
Amino acid. So this, you knew that this would be a question. Wait, I'm like, it's not scrolling.
Oh yeah, because I think this is.
Oh, this is just the question.
I can move my stuff.
So it's S, T, or R.
STY.
I keep doing that. I don't know why.
But more important is what STY has.
Right. So they all have the A is the wrong structure altogether. That's not an amino acid.
Are you sure?
Yeah, because it always has the NH3 on the or NH2 usually, but it's on the bottom.
Oh, I mean, it just drawn
Through
different.
structuring, but none of the amino acids only have like an oxygen as the R group.
Glycine would have an H,
Glycine has the H, yeah.
and you don't.
Oh,
Yeah.
see when I like it's
Mhm.
okay.
But
Right, but glycine is not.
yeah, remember what we talked about. Whenever you see
Yeah.
a something, think something.
Like, are you talking about like the alpha beta?
Um, think about the most common something.
I don't know. Like
Or okay, then think about what STY, what functional groups would they have?
they'd have OH.
Yes.
They'd have CH2. Oh, that.
That's the most common what?
That's the most common electrophile and nucleophile.
Uh, well, how can it be both?
Um, well, it's not, but OH is the base. It's a nucleophile. And then they all have a nucleophile. No. Tyrosine also has an OH. Oh no, they all have an OH.
Yep.
I don't know. It's so hard to read when it's like this. Like I'm thinking of the R groups that I know, and then when it's not.
Well, so so this over here is just the leucine, right? And
Yeah, that's the bridge that you were talking about. And then here we have
so leucine, I believe. But all in all, what we're really looking for is that OH group, right?
uh-huh. And so the fact that none of them have it.
Yeah. And so when you read the this over here, you they're going to ask questions like about kinases and what they do and the types of amino, like that's super high yield. So you're going to see this a lot. So get into the habit of when you read enzyme that phosphorylates amino acid 19, like think kinase. It may not be a kinase, it could be some phosphorylase or something like that.
This
But
one's serine, right? Like the AOH.
mhm.
Okay.
Yeah. So this is just remember
Like
that
for me, the issue wasn't like I remember the STY thing and then I just couldn't. But that's actually really simple, like the OH on the end. I like have a hard time reading the amino acids. Like when I was going through it, I memorized the structure. Like I know what I'm looking for, and then it's the reading it that's difficult for me. But this
yeah,
makes sense.
that's why like I always teach amino acids differently. Like instead
I
of
need to look through your PDF for amino acids tonight. Like I think that it would be really valuable for me.
oh yeah, like I don't have one like specifically just for amino acids, but the way that I teach it is really like I mean I could send a video where I went over it with the student, but the way that I teach it is first look for amino acids that are charged, fully fledged charged ones, and that'll include two of the acidic ones, three of the basic ones.
Right.
And instead of polar, I think partial charge. Because something that's polar just has a partial charge, and that will be STY. And the so the acidic amino acids, glutamic acid, aspartic acid, if you take those and you turn them into amides,
Uh-huh.
aspartic acid will turn
Yeah.
into asparagine, and the glutamic
Glutamine.
acid will be glutamine. Yeah. So that'll be those. So there's five charged, five partial charged, and the rest of the 10 would be like the non-polar.
Non-polar.
So that's why, yeah, I take issue with people who just say memorize the chart because you can know exactly what it looks like, but if you don't know the special, unique properties,
Yeah, for me, when I drew it out, I like highlighted them in different colors. So
yep.
now when I see things like, for example, proline, like I think of it in like a yellow because I know that like that's non-polar.
Yeah,
And so,
yeah. Yeah, on the Jack Weston website, there's like it's not an Anki deck, but it's like on the website, Anki version, on amino acids, and it includes not just like the structure, but unique properties about them. So that could be
mhm.
worthwhile to look at. Because something that is a really weird amino acid is histidine. Because like why do we consider it basic if the side chain is a pK of 6? So that could be like something to look at. So yeah, I would check those out. But yeah, overall, just remember that thing about the most common nucleophile, most common electrophile, and how that relates to phosphate. Um, and
Mhm.
uh, and yeah, so you're doing your next full length tomorrow,
Yeah.
right?
And then when did you want to meet? You said Monday?
Um, Let me see what I can.
Monday's like a little bit challenging for me, especially I wanted to do another full length. I wanted to blind review on Sunday and then do another full length on Monday if I can.
Mhm.
So maybe I can do like Tuesday, like earlier in the day. Does that work for you?
Yeah.
Around one, like we were planning for today.
Sorry, what was the last thing you said?
Could you do like one on Tuesday?
Like you're saying let's meet on Tuesday?
Like 1 o'clock.
Oh, okay.
Or in that vicinity. Like I'm not specifically set on one. I just
Yeah. Um, okay. I put you in for 1 p.m. Eastern, Tuesday, the 28th.
okay.
That works?
Yeah.
Okay.
That's good.
So
Okay.
good luck
I added it to my calendar. So, all right. See you then.
all right, okay. Good luck on your test.
Thank you.
Take care.
Hopefully.